Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we have completed about different types of parts of enzymes, the classification of enzymes, their mode of action, their mechanism of action. Now we are going to start with the catalytic action or catalytic effect of enzyme. That how do enzymes they catalyze a chemical reaction. Enzymes, they are biocatalysts. They help to increase the rate of biological reactions known as metabolic reactions. These enzymes, they have different sites for the attachment of substrate, and that site is known as what? Activation site. We learn about the different mechanism of enzymes action, that is the two main theories which were proposed by Koshland and Frederick Mischer, that is the induced fit theory and the lock and key mechanism respectively. Now enzyme, they speed up the chemical reaction by lowering the activation energy, the amount of energy required to start a reaction. So what exactly is activation energy? See, suppose we are considering two conditions. Condition 1, in which at a given time t, out of total 100 molecules, 40 molecules, they were having the energy equivalent to convert reactant into product. When suppose at a given time t, we are having total 100 reactant molecules, but the product conversion is only 40%. When the 40 molecules of reactant, they are converted into product, rest 60 are unreacted. This is a condition without enzyme. Now, condition 2, at a given time t, in the presence of enzyme, this fraction of unreacted molecules also converts itself into the reacted part. That is, now the efficiency of the reaction becomes 85% into product. So previously, there was an energy barrier which was required to cross by every substrate molecule or maximum substrate molecule to convert reactants into product. But because of the presence of enzyme, that energy molecule or that energy barrier, which is known as what? The activation energy, is overcome by the maximum number of molecules. So a strain is applied on a three-dimensional shape of the substrate upon binding of the substrate to the active site of an enzyme. The moment enzyme reacts with the substrate, the enzyme holds the substrate into its active site. There is a change in the conformation of the active site of enzyme, or we can say there is a strain effect produced after the enzyme substrate complex is formed. So previously, without enzyme, the substrate needs this much amount of energy to be converted into product. But in the presence of enzyme, the enzyme substrate complex, it lowers the energy which is required by maximum number of molecules to convert into product. So this minimum amount of energy which is required by the reactant molecules or the substrate molecules to convert into the product is known as what? Activation energy. Once the chemical bond is weakened, it takes less energy to break the bonds. So once the chemical bonds of the substrate they becomes weakened, they take less energy, they absorb less energy to break their bond. and substrate is converted into the product. In human physiology, when we will study about respiratory system, CO2 gas, it dissolves in water, mean plasma, to form carbonic acid. Without enzyme, the rate of reaction is very slow. But in the presence of carbonic anhydrase enzyme, the rate of reaction, it increases to 150 to 200 folds. Means now the H2CO3, that is carbonic acid formation, increases. So this is the effect of enzyme. Okay. So enzyme helps to convert reactant into the product at lowering the activation energy. Cofactors. What are cofactors? The catalytic activity of an enzyme depends on the presence of small molecules known as cofactors which activate enzyme. These cofactors, they help to activate enzyme. An enzyme without its cofactor is termed as apoenzyme. Okay. So we can say that holoenzyme minus cofactor, it is apoenzyme. Holoenzyme is the total part, that is the proteinaceous part and the non-proteinaceous part. The proteinaceous part is the apoenzyme and the non-proteinaceous part is the cofactor. Okay. So the non-proteinaceous part is cofactor. Apoenzyme plus cofactor is equal to holoenzyme. There are three kinds of cofactors. Cofactors, they can be organic as well as they can be inorganic. Prosthetic groups, coenzymes, and metalines. Prosthetic groups. These compounds are tightly integrated into the enzyme structure by covalent or non-covalent forces. For example, peroxidase, the catalyze breakdown of hydrogen peroxide to water and oxygen. So these compounds are tightly integrated into the enzyme structure by the covalent or non-covalent forces. For example, peroxidase enzyme, it catalyzes the breakdown of hydrogen peroxide into water and oxygen. Heme in hemoglobin is a prosthetic group and it is a part of active site of enzyme. It can be organic or inorganic. Metallines, it can be organic as well as inorganic. Inorganic means metallines. So organic, flavin mononucleotide, flavin adenine dinucleotide, biotin is inorganic. Metals are most common prosthetic groups. Okay. So prosthetic groups and coenzymes, there are two parts. Prosthetic group, they can be organic as well as inorganic. These prosthetic groups, the compounds are tightly integrated into the structure of enzyme by the help of covalent or non-covalent forces. For example, peroxidase catalyzes the reaction of conversion of hydrogen peroxide into H2O and oxygen. Heme in hemoglobin is the prosthetic group and it is a part of the active site of the enzyme. It can be organic as well as inorganic. Inorganic includes metallines. Example, organic is flavin mononucleotide, flavin adenine dinucleotide, and biotin is inorganic. Okay. Biotin is also organic. Inorganic includes metallines. Okay. Like copper, cobalt, magnesium, Mn, zinc, iron. Okay. Coenzymes. Coenzymes, they are organic compounds only. And their association with the apoenzyme and when they combine with apoenzyme is only during the course of catalysis. So coenzymes, they are the organic compounds and their association with apoenzyme is only seen during chemical catalysis. Furthermore, coenzymes serve as cofactors in number of different enzyme catalyzed reactions. Coenzymes, they also serve a catalyzed reaction in terms of cofactors. The essential chemical components of many coenzymes are vitamins. For example, coenzyme nicotinamide adenine dinucleotide NAD and NADP contain vitamin niacin. Okay, so nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate contain vitamin niacin. Okay, Number of enzymes require metal ions for their activity. This activity forms coordination bonds with side chains at active site and at the same time forms one or more coordination bonds with the substrate. Number of enzymes required metal ions for their reactivity. The activity forms coordination bond with the side chains at the active site and at the same time forms one or more coordination bond with the substrate. For example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Zinc is a cofactor for the enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that cofactors play a crucial role in the catalytic activity of enzyme. Okay, because most of the cofactors, they are either organic or inorganic. They help to make the substrate compatible according to the enzyme. Okay, so coenzymes, they are organic and their association with apoenzyme is very important, and it is only seen during the course of catalysis. Coenzymes serve as cofactors in a number of different enzyme catalyzed reaction. The chemical components of many coenzymes are vitamins like coenzyme nicotinamide, adenine dinucleotide, and dinucleotide phosphate. They serve as the energy carriers. They are vitamin rich, in which vitamin is present? Niacin. Okay. Then the riboflavins, flavin, adenine dinucleotide flavins, they all contains the vitamin B2, vitamin B1, okay, thiamine and riboflavin. Catalytic activity is lost when the cofactor is removed, as the cofactor play a crucial role in the catalytic activity of enzyme. What are the different qualitative tests for some important biomolecules? This is very important, as you have to study about the different qualitative tests which are done to complete the diagnosis of the biomolecules present in either in blood or either in urine. Okay. Suppose a person is suffering from some disease like glycosuria. Glycosuria is a condition in which and there is an presence of carbohydrates seen in urine. So presence of glucose, simple sugars like glucose are seen in urine. So their presence is tested by either Felling's test for glucose and fructose. They are grape juice, mean glucose is a grape sugar, and fructose is a fruit sugar. Okay, contains glucose and fructose. Their presence can be tested by Felling's test and Benedict's reagent. Okay, so what you have to do for Felling's test, there is a solution Felling A and Felling B. Felling A is aqueous copper sulfate and Felling B is sodium potassium tartrate, Rochley salt. Potassium tartrate. Understood? They are mixed in equal amount. Now, take 5 ml of fruit juice in the test tube, add equal quantity of Felling A and Felling B, boil the solution, a brick red precipitate of cuprous oxide brick red precipitate indicates the presence of glucose or fructose in the fruit juice. Okay. Fructose gives red color with Selvanov's reagent. Selvanov's reagent. Selvanov's reagent is resorcinol plus concentrated HCl. Okay. While glucose gives no coloration. with Selvanov's VG. Okay, there are many more tests like glucose. It gets or undergoes charring only on heating by the action of concentrated H2SO4, while fructose chars in cold. Charring is done by adding concentrated H2SO4. Benedict solution is also done. The Benedict solution, on treating with the Benedict solution, green, red, yellow, or rust brown precipitate is formed. Is formed. Okay. Now, sucrose doesn't give Felling's test. It is first hydrolyzed by conc H2SO4, concentrated H2SO4. Okay. So on treating with H2SO4 concentrated, the sucrose, it gets hydrolyzed. 5 ml of sucrose solution with few drops of concentrated H2SO4. It is then neutralized with NaOH to maintain the pH. Then we will boil again by adding Felling solution. Now it will give. Okay. Because sucrose is basically, it will not give the Felling's test as it is not having the free aldehydic or ketonic group. Okay. Now test for fats and oils or lipids. So we can use 0.1% Sudan third solution made in alcohol, okay, which gives the reddish color. Okay, the presence of reddish color, it helps in the diagnosis of fats and oils or lipids in the given mixture or given sample. We can also test for glycerol by mixing 1 ml of 1 ml of 1% copper sulfate solution and add 5 drops of glycerol. Blue color is obtained. To test for glycerol can also be done. One ml of 1% CuSO4 solution and 5 drops of glycerol. Okay. Blue color is obtained. So these are the tests for fats or oils or lipids. First, you can use 0.1% of Sudan third solution, which will give the reddish color, and then the test for glycerol is done by adding 1 ml of 1% copper sulfate solution and 5 drops of glycerol. Blue color is obtained. Then the test for proteins, which includes xanthoprotein test. Xanthoprotein test, we will take 5 ml of protein sample, added with 2 ml of concentrated HNO3. Heat it, boil it, a yellow color appears after heating. Yellow color appears. Then we will cool it and add excess of 20% NH4OH or NaOH. And add 20% of NaOH or ammonium peroxide. Orange color is formed. It is due to the orange color is formed. It is due to the nitration of, it is due to the nitration of phenolic groups attached to the side chains of aromatic amino acids. So aromatic amino acids can be detected by the xanthoprotein test. You can see the phenol, mean the OH group aromatic alcohol. Okay, it gets nitrated. The orange color, orange yellowish color. This is only for aromatic amino acids. Like tryptophan, tyrosine. Then the biuret test. The biuret test is detection of the peptide bond. 3 ml of protein sample is added with 1 ml of 40% NaOH. And add few drops of 5% copper sulfate. Then we will shake it and keep it for 15 minutes at room temperature. A violet pink color appears. Okay, understood? The appearance of violet pink color, it gradually changes to blue and purple. You can see in the test tube. Okay, the violet pink color gradually changes to blue and purple. Okay, so this test, biuret test, is also for proteins. It helps to predict the peptide linkage. The violet colored complex is known as what? Biuret. The test for amino acids, ninhydrin test is the best method to detect amino acids. Most of the amino acids give purple color with ninhydrin. Okay? But tyrosine, phenylalanine, aspartic acid give blue color. Except tyrosine, phenylalanine, and aspartic acid. Okay? They give blue color. Tryptophan produces olive brown. And proline gives yellow color. Now, test for urine. So the urine sample is tested for confirming the presence of urea, uric acid, creatinine, minerals, sugar, and albumin protein. test for urea. Urea, when decomposed, liberates ammonia. Microbial, liberates ammonia. Okay, and forms a biurate. Biurate test. For detection of sugar in urine. Benedict test. Okay. For detection of albumin, both in blood and urine. So I think, my dear students, we have completed the different tests as well, like xanthoprotein test, biurate test, Pavlov's reagent. So please revise everything, especially these tests. We will be meeting the next turn to start the new chapter, that is human physiology. Before that, we will conclude with good number of questions from this chapter. Till then, bye bye, take care, stay safe, stay healthy.